Today on the Lab Report, we'll learn how human anatomy is similar to the anatomy of many organisms. That humans developed from an embryo with a temporary tail and resemble the embryos of other organisms. And we'll learn that scientists look at fossils to see how anatomy of organisms has changed over time. Hey, I'm Dr. Jeff, and today we're learning about comparative anatomy. That means we'll be looking at structures like bones and comparing them between different organisms. Adam, Caroline, say hello to Randy, our male human skeleton.
Hey, Randy.
Hey.
Sup? Today, Randy is going to help us look at the anatomy of a human. What do you remember about the skeletal system?
It consists of all the bones and joints in our bodies, which do things like provide a structure and protect our organs.
And there's something like 200 bones in the human body.
206 to be exact, but who's counting?
Since you mentioned that Randy is a guy skeleton, do girl skeletons look the same?
Great question. Let's ask Barbara, a female human skeleton. Here she comes now.
Hey, Barbara.
Howdy.
Perfect. Take a close look. How many differences in their bones can you notice?
I don't notice anything different. They look exactly the same.
That's right. That's because they are the same. Males and females have the same bones arranged in the same way.
But don't boys usually end up taller than girls when they're fully grown? I don't see a difference at all.
On average, yes. Also, men's shoulder bones are slightly wider, and women's hip bones tend to be slightly bigger. But the number of bones and their arrangement is the same. For example, let's compare the rib cages.
Well, Barbara has 12 bones in her rib cage, and so does Randy.
And Randy has 24 bones in his spine, and so does Barbara.
Exactly. Now let's take a look at the anatomy of a human arm. To study the arm bones, we will use a human arm. Okay, fine. I volunteer Barbara. This won't hurt one bit. Excuse me.
Ow.
Thank you.
Good thing Barbara doesn't have a nervous system.
On the table, we have a dog leg, a bat wing, and a dolphin flipper, and now a human arm. Let's compare them.
Well, the human arm has a big bone connected to two bones connected to a bunch of smaller bones connected to fingers. And this dog leg has a big bone connected to two bones connected to a bunch of smaller bones connected to fingers.
Yeah, this bat wing has the same pattern. One bone connected to two bones, connected to a bunch of little bones, connected to fingers at the end.
And this dolphin flipper has the same pattern. One big bone connected to two bones, connected to a bunch of bones, connected to finger-like bones.
Excellent. We see the same pattern in the arms of many animals, like elephants, turtles, frogs, and more.
So is this pattern just seen in arms and legs?
Good question. Let's find out by exploring more of the anatomy.
Whoa, is that a hawk?
It is, and this is a Komodo dragon, the largest lizard on planet Earth. And you know Randy, of course.
Hey, Randy.
Sup? Let's compare these three skeletons from very different animals. What do they have in common?
Well, they all seem to have the same basic pattern. There's a skull, which connects to the spine, which connects to the shoulder blade, and then to the limbs.
And the limbs have the same pattern we saw before.
And their legs connect to the hip bone, which connect to the spine as well. So they all look very different, obviously, but they have the same basic arrangement of bones.
And look, all three spines end in a tail.
What? Randy doesn't have a tail?
Well, looks like he does. It's right there.
Caroline is right. Those bones in a human are called the tailbone. We all have one, and you're probably sitting on it right now.
What? Why would we have a tailbone but no tail?
Well, we all had a tail when we were very little. You remember?
Definitely not me.
Definitely you did. You don't remember because you were only this big when you had a tail, and it was only there for a few weeks.
Why would we grow a tail for only a few weeks?
To answer that, let's take a look at the development of animals in the first few weeks of growth. These are baby chicks.
They hatched just a few days ago.
They're so cute.
But before they hatched, they developed inside an egg. Here we have samples from the development of a chicken at various stages. Let's take a look.
Weird. They don't look anything like chicks.
That's because organisms look very different as they develop. These are the single cells from which a turtle, chicken, rabbit, and human develop. They all start off as single cells. Now, let's fast forward a few weeks.
Weird. They're all developing the same way, and they even have tails.
And they all have little ripples under their heads. What is that?
Those are called pharyngeal slits, which in fish develop into gills.
That's crazy. So when chicks and humans are embryos, we have parts similar to gills and a tail?
Yes, the tail goes away as we grow, but we still have a few bones from that tail in our tail bones.
So all of these animals, from chickens to frogs to humans, begin their development in the same basic way.
Exactly. Comparing embryos reveals similarities not seen in the fully grown anatomy.
So our bone structure matches so many animals, and we even develop the same way as fish and chickens. But why? Why is it that we're so similar?
Is it a coincidence, or are we all related somehow?
Excellent questions. To answer them, we'll have to look at fossils. Scientists compare current living things to fossils to see how their anatomy has changed over time. For example, here is the skull of a modern horse. And these are the skulls from horses that lived 10 million years ago, 20 million years ago, and about 35 million years ago. What do you notice?
It looks like its shape and size changes over time.
Exactly. Now take a look at the fossilized leg bones of those same animals. What do you notice there?
They also change over time. The oldest one has 3 toes, then they become smaller in the next one, and then they disappear. Today's horses only have 1 toe.
Yes, scientists observe these kinds of anatomical changes with many different animals. Let me show you another one. Here is a modern whale skull. Notice the blowhole is at the top. And here's a whale skull from 40 million years ago, 45 million, and about 50 million years ago.
It looks like the position of the blowhole changes in each fossil.
That's right. The change in living things over generations is called evolution. We can see evolutionary changes in the fossil record of horses, whales, snakes, birds, and much more. The fossil record is such a big topic that we have a whole separate lesson to explore it in depth. The lesson is called the fossil record.
Now, let's explore the kinds of careers you can have studying comparative anatomy.
A developmental biologist is a type of scientist that studies how living things can grow from a single cell to become a highly complex organism. Things can go wrong in this complex process, and developmental biologists try to understand what went wrong and what caused it. For example, certain substances can cause errors in the process of development, leading to birth defects. These substances include alcohol and certain medications used to treat diabetes. It's because of the work of developmental biologists that mothers know not to take these substances when pregnant. A veterinarian is a type of doctor that uses comparative anatomy to treat animals. Their patients can be dogs, cows, birds, snakes, and even lions. Understanding shared anatomy can help vets take care of any animal that needs help. For example, if a vet hasn't treated a rare Central American wood turtle before, but they have treated a common painted turtle, they can use their knowledge of comparative anatomy to help the turtle in need.
Today, I'll show you how to search for bones and compare your skeleton to that of a rodent. To do this, you will need tweezers, a popsicle stick, a white piece of paper, a sterilized owl pellet, and bone identification charts. Since owls don't have teeth, they swallow their prey whole. Any undigested bones and fur are regurgitated as a pellet, making owl pellets a great place to look for bones of rodents and small birds. You can find owl pellets online for about $10, and they usually come with a chart to help you identify the bones. First, lay out the white piece of paper and open your sterilized owl pellet. Next, use the wooden stick to gently break apart the owl pellet. Next, use the tweezers to pick up and examine the bones over the paper. Now, use the bone identification charts to sort and arrange the bones by animal, usually a rodent, shrew, mole, or small bird. This looks like a small tail from a rat. Cool. And this looks like the head of a mole. Ugh. Once sorted, you can compare the bones you found to human bones. We should be able to see many of the shared bone structures we learned about today. Try it yourself.
Thanks, Adam. Now, let's review.
Today, we learned that the anatomy of the human body is similar to many other organisms.
Humans developed from an embryo with a temporary tail and resemble the embryos of other organisms.
And scientists look at fossils to see how the anatomy of the organisms have changed over time.
Join us next time on the Lab Report. I'm Dr. Jeff.
I'm Adam.
I'm Caroline.
And remember, always question, always wonder.